welcome Professor Xiao.
Good afternoon, everyone. My name is Yu-Yuan Hsiao. I'm from the Institute of Molecular Medicine and Bioengineering in NYCU. It's a great pleasure to be here to show you our current results. And today I would like to talk about a unique allosteric inhibition mechanism of targeting covalent inhibitor, TCIs, on an antiviral drug target, NPexo nucleus. The TCIs specifically interact to a residue that is not located at the active site or substrate binding site, and block the nucleus activity of NPexo by interfering the protein dynamic. And later I will show you the detail. At first, I would like to give a very brief introduction of my lab. My lab is a structural biology laboratory. We are focused on the catalytic properties, substrate selection mechanism, and cellular functions of nucleic acid binding proteins. Currently, we are engaged in the study of some nucleus involved in immune regulation, such as TREX1, SMHD1, and RSH2. On the other hand, when the cell under the genotoxic stress, TREX1 and its working partner dislocate into the nucleus and participate in the DNA repair pathways. So we also very interested in their rules in response to the genotoxic stress. Interestingly, most proteins are drug target. So we also aim to use the structure-based drug design to find the inhibitor and apply it in cancer immunotherapy and anti-cancer drug development. And the most exciting target is TREX1. TREX1 is belong to DEDDh exonuclease superfamily. The family is a very large protein family, including over 13,000 members, and widely exists from bacteria to mammalian cell and virus. And some of them are drug targets. For example, the inhibitor of TREX1 can apply in cancer immunotherapy, and the inhibitor of TREX2 can apply in the treatment of skin disease, psoriasis. And the inhibitor of NSP14 can be applied against viral infection to SARS-CoV-2. In 2016, we have determined a complex structure of DEDDh exonuclease and inhibitors. As you can see here, the green one is the apoprotein structure, and the blue one is inhibitor bond structures. As you can see here, after the inhibitor binding, the most important residue, the journal base histidine, is moving out away the active site, and then the enzyme activity is inhibited. So this is a very interesting inhibition mechanisms. As we know, to understand the inhibition mechanism of different types of inhibitor, it's very useful to help us to design next generation drugs. So we are also very interested in the in revealing the inhibition mechanism of many different types of inhibitor. And today we want to focus on the NPexo nucleus and show you a unique inhibition mechanism on the NPexo nucleus. NPexo nucleus is an enzyme in the Lassa fever virus. According to the WHO statistics, the Lassa fever virus infects about 300,000 to 500,000 people in West Africa. And until now, there are no effective treatment or vaccine against this virus. And the NPexo nucleus is a promising target against the Lassa fever virus, which is composed by the N-terminal domain and the C-terminal nucleus domain. And the C-terminal nucleus domain belongs to the DEDDh exonuclease superfamily and responsible to degrade the double-strand RNA in the virus. And why NPexo nucleus is a promising target? It is because the double-strand RNA is a kind of pathogen-associated molecular patterns, PAMP. The PAMP can detect by our cytosolic nucleic acid sensor and activate the innate immune system for viral killing. When the NPexo nucleus degrade the double-strand RNA and the pathway is blocked, so we cannot kill the virus by innate immunity. If we can develop an inhibitor of NPexo, we can restart the innate immunity to kill the virus. So NPexo is a promising drug target against Lassa fever virus. And we also want to use the NPexo nucleus as a model system to study about the inhibition mechanism of TCIs in the DEDDh exonuclease. The full name of the TCIs is targeted covalent inhibitors. TCIs account for 30% of the marketed therapeutics and be recognized as a promising strategy against complicated diseases like cancer or viral infections. The most famous example is the ARS1620. This drug can specifically interact to the cysteine residue in the KRAS mutant and induced cancer tumor regressed with higher potency and specificity. So this is a very successful case. On the other hand, there are still some disadvantages of TCIs, like the off-target toxicity. So to understand the inhibition mechanism of TCIs is very important and very useful for next generation TCI as inhibitor development. And today, I want to use PCMP and PCMB, the two typical TCIs as an example to study about the inhibition mechanism of TCIs. PCMPs and PCMB are typical TCIs that can inhibit a lot of enzymes, including kinase, preoxidase, or phospholipase, or isomerase. However, some of the inhibition mechanisms remain unclear. The crystal structure clearly shows that PCMPs can react to the SH group to form a mercury sulfate band to the target protein. If the cysteine residue is located at the active site or substrate binding site, so the inhibition is very reasonable. But in some other case, as you can see here, the cysteine is not located at active site, and the binding of PCMP also not affect the overall structure of the target protein, and the inhibition mechanism remain unclear. So our specific aim is we want to know the inhibition mechanism of the PCMPs and PCMP on the target enzyme. In 2016, we have identified the PCMPs is inhibitor of some DEDDh exonuclease, like CRN4 and RNST. So now we want to test the inhibition of other DEDDh exonuclease, especially the drug target, like JACS1 and ASP40. So similarly, we can see the inhibition. When we add the PCMPs and PCMP into the reaction, most of the substrate cannot be degraded by the JACS1 and ASP40. So we can see the inhibition effect. Similarly, in the NPexo case, we also can observe the inhibition effect in a dose-dependent manner. Because I am a structural biologist, So I want to use the structural approach to revealing the inhibition mechanism. So I determined several structures, including wild type, mutated NPexo nucleus in complex with PCM-PS or PCM-B. And here is the apolipoprotein structure of the nucleus domain of NPexo. And here is the overall structure. And the active site residue is very similar to other DEDDh exonuclease. And another important region is here. Here is the link binding site. The link binding site is very important for stabilize the overall structure of the nucleus domain, and also associated with substrate binding and specificity of NPexo. And here is the structure of NPexo PCM-BS complex. As you can see here, the yellow one is the complex structure. PCM-BS react to the cysteine 409 residue in here. And this site is different from the active site and zinc binding site. And we try to do the structure superposition. The yellow one is complex structure, and the green one is the apolipoprotein structure. As you can see here, the PCM-PS binding is not effect of the orientation of active site residue and the ligand binding site residues, and also not affect the overall structures of NPexo. And in another complex structure, in the structure of NPexo PCM-B complex, PCM-B is another TCI, we can see the similar phenomena. The PCM-B also react to the cysteine 409 residue, and also not affect the overall structure and the orientation of the active site residue and zinc binding site residue. Even though we cannot observe a significant structural feature for revealing the inhibition mechanism, but we still can understand that this residue, cysteine 409, is very important for inhibition. If we mutate it into the element, the inhibition may be disrupted. Unfortunately, when we generate the NPexo mutant, we find that this mutant still can be inhibited by PCM-BS or PCM-B. So we guessed maybe there are another cysteine residue can react by PCM-B and PCM-BS. So in here is another crystal structure of NPexo nucleus, a mutated NPexo nucleus in complex with PCM-BS complex. So you can see the PCM-BS target to another cysteine residue, a cysteine 461, and it is also not located at the active site or the zinc binding site. And it only affects a local conformational change and also not affect the overall structure. And it also not affect the orientation of the active site residue and zinc binding site residue. In order to check our structural observation is correct, so we perform the mass spectrometry experiment. Compared to the mutated NPexo, the wild-type NPexo have one additional peak. It means that the cysteine residue is very important for PCM-B targeting. And also we can see there are two peaks. It means that there are two cysteine can target by PCM-BS. So the result is very consistent of our structure observations. And then go back to our specific aim. How PCM-BS inhibited the NPexo? So we guessed maybe the binding site of PCM-BS is located at the substrate binding site. Unfortunately, we are wrong again. So as you can see here is the substrate binding site, and cysteine is here. It's very different. So another possible way for inhibition may be PCM-B affect the dynamical punctuation of a target protein and then inhibit the activity. So we cooperate with Professor Zhu Ziwei and do some ND simulation. And here is the result of ND simulation. As you can see here, you can clearly see the motion of the two proteins very different, especially in the blue and red region. The two regions are essential for its activity. So I know it's very difficult to feel how the difference between the structure. So I try to show you a figure to explain the difference. As you can see here in the apo protein structure, there are several hydrogen bonds to make the secondary structural coupling between several secondary structures. And after the ND summation, we can see the hydrogen bond is still, and those structures are coupled together. But in the PS bond NPexo, you can see after ND simulation, the coupling is disrupted. It means that the PCMP indeed affect the dynamic of NPexo. Here is another example. Here is another coupling between the alpha-3 and alpha-8 structure. But in the NPexo PCMP complex, after ND simulation, the coupling is disappeared. And why? The reason is the SO3 group of the PCMP can interact with the arginine residue and pull the alpha-8 to move in close to the beta-3. And then the coupling is disrupted, so the interaction is disappeared. So we can conclude that the local perturbation due to the PCMP propagate further by intra-protein coupling. The dynamic strongly affect the nucleus activity and protein conformation of the target protein. And here is another simulation result. Shows very similar result. As you can see here, so after we did the PCMP in the structure, so after ND simulation, there are also some coupling is disrupted. In order to check our ND simulation is correct, so we try to perform the binding assay to check. If the PCMP can affect the dynamic of the target protein, the substrate binding ability should be also affected. So we try to use the Joe-Shifter experiment to check. So as you can see here, when we did PCMP and PCMP into the reaction, the complex band is disappeared. It means that PCMP and PCMPs also affect the binding ability of target protein. So we can conclude that this experiment indirect proof our hypothesis. In summarize our result, we try to use the structure and biochemical and bioinformatic approach to reveal a unique allosteric inhibition mechanism. And as you can see, the two PCMP molecule can interact with the allosteric side and influence the protein dynamic and then inhibit the target protein. So here is a take-home message. I guess the inhibition sometimes not all related to the active site or the overall structure. The dynamic also is a very important point for inhibition. So finally, I will say thank you to my financial support. The first one is most, and the second one is IDS2B Center from the MOE. I also want to thank the SRC to help us collect the X-ray diffraction data. We also thank to our collaborator, Professor Zhu Ziwei. And this work is done by Guang Wei and Youzi and Rongyu and Caiyuan. And here is the QR code is can link it to our website. And here is our new YouTube channel. If you are interested in our research, you can just screen the QR code to see the videos. And I would like to stop here, and thank you very much. I'm happy to take questions.